Delineation of α-synuclein structure using Förster Resonance Energy Transfer. Lewy body disease is an umbrella term that refers to both Parkinson's disease and Lewy body dementia. Together, they affect more than 5 million people worldwide. As its name suggests, this disease is characterized by Lewy bodies, which are abnormal aggregate of protein in neurons, primarily composed of α-synuclein protein. Past studies using traditional methods reveal that α-synuclein is an intrinsically disordered protein, lacking a distinct three-dimensional structure. Under pathological condition, unfolded monomers of α-synuclein interact to form dimers, which grow slowly to generate oligomers that eventually convert to fibrils. The accumulation of these fibrils result in Lewy bodies. Scientists have recently incorporated Förster Resonance Energy Transfer, or FRET, in a double-labeled α-synuclein to monitor the intermolecular conformational change under different buffer conditions. FRET involves two fluorophores, a donor molecule and an acceptor molecule. First, electrons of the donor molecule are excited by light energy and undergo relaxation. When the emission spectrum of the donor overlaps with the absorbance spectrum of the acceptor, the energy can be transferred through non-radiative dipole-dipole interaction. In the following graph, green curve represents the emission spectrum of the donor fluorophore, while the red curve represents the absorbance spectrum of the acceptor fluorophore. The shaded region is the overlap. When a protein is labeled with both fluorophores, the emission spectrum is quenched and the energy is transferred to acceptor through resonance. The efficiency of quantum energy transfer is analogous to a person throwing a ball to another person standing at a certain distance. The further your partner fluorophore stands, the less likely you will successfully throw energy to him. The equation between FRET efficiency and the distance is: FRET efficiency is experimentally obtained using fluorometer data, and the distance r is calculated. Förster distance, or r0, is the distance at which transfer efficiency is 50%, which can be calculated by using different properties of the fluorophores and the medium. FRET is highly sensitive to the distance between the donor and the acceptor, indicated by r to the 6th power in the denominator. A typical efficiency versus distance graph of FRET looks like this. FRET has several advantages over other techniques in studying the α-synuclein conformation. First, its optimal distance for energy transfer matches the size of the protein. Second, low concentrations of proteins is sufficient to monitor FRET. Third, energy transfer is usually not influenced by the presence of other biomolecules. It is true that the limitation of FRET lies in its inability to produce single correct image of the protein. However, it greatly reduces the number of possible options that can be further improved using other imaging techniques. A recent study conducted by Dr. Peterson uses thioamide as an acceptor fluorophore and p-cyanophenylalanine, or CNF, as a donor fluorophore. An α-synuclein protein is labeled with CNF and thioamide. The double-labeled protein is then diluted in urea and trimethylamine, or TMAO buffer solutions, which are known to denature and compact α-synuclein respectively. To calculate the FRET distance between the two fluorophores, the fluorescence of CNF thioamide labeled α-synuclein F-thio and a CNF labeled control F-oxo is measured. In the normalized emission spectrum of both proteins, F-oxo and F-thio values are obtained. Then, quenching efficiency is calculated using these spectroscopy data. Finally, the actual separation distance, or FRET, can be deduced from the following equation, using the previously calculated quenching efficiency. In order to acquire information of the overall structure change, this experiment is repeated with fluorophore pairs and different amino acid residues. After obtaining distance library of several residue pairs, a simulation program is run in order to deduce possible structures of α-synuclein. The usage of thioamide as a quenching molecule is significant in that the modification can be made in various protein sites. Biomolecules containing peptide bonds have numerous side chain oxoamides that can be easily converted to thioamides. Therefore, with minimal modification, thioamide can function as a versatile FRET probe. This opens up another possibility in which FRET can be developed to visualize the conformational changes of various intrinsically disordered proteins.